All right, at the end here of the unit, we're going to cover some of the most difficult concepts that are in this unit. And this first one goes back to what was discovered by Thomas Hunt Morgan. If you'll recall, he's the one that found this white-eyed mutation, this white-eyed male fly, and then used it in genetic experimentation. And he was the first to demonstrate what we now call gene linkage. The fact that the gene for eye color that was mutated to produce this white eye is on the X chromosome. And I added this image. You don't have this in your discussion guide, but I wanted to show you the difference between the X chromosome and the Y chromosome in humans. So this is a human X and Y chromosome scanning electron micrographs, and they've been colorized. But you can see that the Y chromosome is much smaller than the X chromosome, and it has many fewer genes and not the same genes. So a sex-linked trait is a trait where the gene is on the X chromosome, because the Y chromosome doesn't have those genes that are sex-linked. Now that specifically is sex linkage, but we're also going to be considering gene linkage in general. And what that refers to is when genes are very close together on the same chromosome, they're more likely to be inherited together. But we're going to use this sex-linked example because it's very clear it's a little easier to understand what we're talking about when we're talking about sex linkage. So there's the female with red eyes and a male with white eyes. White eyes is a mutation, and Thomas Hunt Morgan discovered a lot of mutations and therefore genes. Genes, the way that a lot of genes were discovered is because they mutate, and we discover those genes once we find a mutant for those genes. So the X chromosome has this eye color gene, and that's what we refer to as a locus, a location of a gene. Once we determine the location of the gene, that's referred to as the gene locus. It's just short for location, basically. And that was discovered by Thomas Hunt Morgan, of course. Due to its location, this type of gene linkage, inheritance trait is known as sex-linked. So in other words, what I'm trying to say here with all these slashes is that this is sex linkage, it's sex-linked inheritance, and it's a sex-linked trait. So you can refer to what's going on here in all those different ways. So let's take a look at a Punnett square, and what Thomas Hunt Morgan did when he discovered this mutation was go ahead and repeat Mendel's experimentation. So if you'll recall, one of the other important things that Morgan discovered is that the principles that were discovered by Gregor Mendel apply to all living things, not just pea plants. So we're going to go from the parental generation to the F1 and from the F1 to the F2, and then we're going to take a look at how it is that you can end up with a white-eyed female, because it is possible. So starting with the parental cross, note that the symbols that you have to use include the symbols for the X and Y chromosome. And then the allele symbols are superscripts on those chromosomes. But you'll notice right off the bat here that it's only on the X chromosome. It's not on the Y chromosome, because this gene doesn't exist on the Y chromosome. It is only on the X chromosome. That's what makes it sex-linked. So this is our white-eyed male. White eyes is recessive to red, so we're using a lowercase r for white. The female then is homozygous dominant red, which is also referred to as wild, the wild character, because normally fruit fly eyes are red. So then if we solve the Punnett square, we find that we have a female, XX is female, so we have a female that is heterozygous, male that has red eyes, or males that have red eyes, because again, one female can lay 500 eggs, so if you just cross only two flies, you're going to get a lot of offspring. But what Thomas Hunt Morgan did was cross a lot of flies and get a lot of offspring. Here we see a male that has red eyes and another female that has red eyes. But notice that the females are carriers of the recessive allele for white. All the flies have red eyes, so the phenotypic ratio here is all red, or 4 to 0, And that's what you expect in the F1 generation, so this wasn't a surprise. And then Morgan took flies from the F1 generation and crossed them to create the F2 generation. So here's our male with red eyes and a female, because both all the males in the F1 generation were the same, have the same genotype. They're all what we call hemizygous. They're hemizygous red. Hemi means half, because they have half the number of alleles that you normally have for any gene. And that's the way it is for these sex-linked traits. The male is going to be hemizygous, whether it be dominant, hemizygous dominant, or hemizygous recessive. There is not an allele on the Y chromosome, so males are always going to be hemizygous for sex-linked traits, and that explains why they're more likely to show a sex-linked trait. So here's our male from the F1 generation, and here's our female from the F1 generation. Likewise, females all have the same genotype. And then we solve the Punnett square, and we find that 1/4 of the females, or 1/4 of the flies, are females with red eyes, homozygous red. 1/4 are males with red eyes. 1/4 are males with white eyes. So here we see the white eye trait reappear, just like his grandfather. And then we have 1/4 female red eyes, but they're carriers. But the phenotypic ratio here is, just like the phenotypic ratio in the F1 generation, all red, wasn't surprising. Same thing in the F2 generation. Not surprising, because that's what we normally see in the F2 generation, is a 3-to-1 phenotypic ratio. But when Morgan looked more closely at what was going on here, he saw that only the males had white eyes. And that was the indication that something strange was going on, and he sat down and figured out that that can only be explained by that gene being on the X chromosome. So he was the first to demonstrate experimentally that genes are on chromosomes. So that's one of the important things that he discovered here, along with the whole concept of sex linkage and this idea of gene linkage in general. So there's our one affected male, and none of the females are affected. However, females can be carriers. So now let's take the affected male and cross him with a carrier female. And again, the whole reason here is I just want to demonstrate that it's possible to get a white-eyed female. It's just less likely. So 1/4 female carrier, 1/4 male with red eyes, 1/4 male with white eyes, and 1/4 female with red eyes. But she has to be homozygous, recessive, to have red eyes. The male only has to be hemizygous, recessive, to have red eyes. And that's how sex linkage works. So here are our affected flies in this generation. Or I should say in these offspring, because I don't want to imply that this is the F3 generation. This does not follow Mendel's experimental design, so it's not the F3 generation. All right, so these predicted results match what Thomas Morgan saw, so it demonstrates linkage because inheritance of white eyes depends on gender. Males are hemizygous and show the phenotype of only one allele. So whatever one allele they have, that's the phenotype they're going to show. And this eye color gene is linked to all the other genes on the X chromosome. So again, the idea of linkage is that if you have genes that are on the same chromosome are more likely to be inherited together, which seems to be a violation of Mendel's principle of independent assortment, because Mendel didn't observe any linkage in his results. All the genes that he looked at assorted independently. We now know that there are two ways to explain that. Either the genes he was investigating are on separate chromosomes, and therefore they're not linked, they're not on the same chromosome, so there's no chance of them being linked, or there's something that is separating genes that are on the same chromosome so that they appear to assort independently. And again, so we now know what that is crossing over. Linked genes are not always inherited together, and they often separate through recombination by crossing over. However, when it comes to sex linkage, there's little to no crossing over between the X and Y chromosomes. So again, they have different genes. The genes that are on the X chromosome, there are many more genes on the X chromosome than the Y chromosome. So there's very little to no crossing over between the X and Y chromosome. All right, so it turns out that we can use crossover frequencies or recombination frequencies to find out how far apart genes are on the same chromosome. So just a little review here. Drosophila has 8 chromosomes, Drosophila being fruit flies, 4 homologous pairs. What we're looking at here is a genetic map or a gene map or a chromosome map of chromosome 2. Number 2. So this is not the X chromosome, this is chromosome number 2. And these genes were first identified by mutation. These are some of the other mutations that Thomas Hunt Morgan found and that have been found since. Crossover or recombination frequency describes how often genes on the same chromosome are separated by crossing over. How often, or what is the how frequently. So that's why we call it frequency. And it turns out that it's directly proportional to the distance separating them on the chromosome. So the idea here is that we can use crossover or recombination frequencies to determine how far apart genes are on a chromosome. So Morgan and his students that developed this have set one map unit, what we now call a centimorgan, or can be called a centimorgan in honor of Thomas Hunt Morgan. One map unit is the equivalent to a recombination frequency of 1%. So what that means is that if 2 genes on the same chromosome are separated by crossing over 1% of the time, then they are one map unit or one centimorgan apart from each other on that chromosome. So the idea is that the closer genes are on the same chromosome, the less likely they are to be separated by crossing over. So the lower the recombination frequency, the closer the distance between genes, or another way of saying that is the more strongly they are linked, the stronger the link between them. So in this example, you'll notice that the black and the purple genes are the closest together of all the genes that are being shown in this chromosome map. And so they are the strongest linked here on this map. They are only 6 map units apart, or 6 centimorgans apart. That means that they are separated only 6% of the time by crossing over. Their crossover frequency is only 6%, which is a very low percentage of being separated. So that indicates that they are very close together on the same chromosome. And that's the opposite of independent assortment. So these genes appear to assort independently only 6% of the time. The rest of the time, they are linked, and they're inherited together, which would have blown Mendel's mind because that violates the whole idea of independent assortment. All right, so the way that they constructed this map, they set this Aristillus mutation to 0, and let's just run through some of these mutations to see what they're mutations of. So Aristillus is short antennae, right? So it's an antenna mutation. Dumpy is a wing mutation. So you'll notice the wings are dumpy. Then these fruit flies really can't fly. Dox, short legs. So the comparison here is shorter legs. Black refers to the body color, which is normally gray, but black is much darker. Purple is eye color. And so we already saw that there's an eye color gene on the X chromosome responsible for white eyes, and that's a sex-linked trait. But there are other eye color genes on other chromosomes, which means that eye color is polygenic. It's actually controlled by more than one gene. And we see the same thing here with wings. The shape of the wings is controlled by more than one gene, so they have polygenic traits. And then there's vestigial, which is another wing mutation, and again, they can't fly. Curved is another wing mutation, so there are a lot of wing mutations, as you can see. And then brown is another eye mutation, eye color mutation, brown eyes instead of red. And you'll notice that the crossover frequency or recombination frequency for Aristillus here is 0. That's because they set it at 0, and they did that to compare. They knew it was near the end of the chromosome, and so they set Aristillus to 0, so that and then they compared the distance that the other genes were from Aristillus. So all of these numbers are in comparison to Aristillus. In other words, they all represent the recombination frequency of the gene to Aristillus. In other words, how frequently are they separated from Aristillus? So this first dumpy gene here separates from Aristillus 13% of the time. So it's 13 map units or centimorgans from Aristillus. Dox separates from Aristillus or is separated by crossing over 31% of the time. So it's 31 map units from Aristillus, and so on. And when it comes to these crossover frequencies and the whole concept of independent assortment, genes that show recombination frequencies greater than or equal to 50% are considered to assort independently and not be linked. So on this chromosome map, that means that purple and vestigial and curved and brown all assort independent of the Aristillus gene. They're all above 50%. So they are considered to assort independently. In other words, they might as well be on another chromosome. They cross over so often that they might as well be on a separate chromosome. So even though they are on the same chromosome as Aristillus, they appear to assort independent from Aristillus. And that's really the exception, not the rule. Most genes appear to assort independently. So purple and above, meaning purple and the rest of these that have a higher frequency than purple, appear to assort independently from Aristillus. So how do we find these crossover frequencies or recombination frequencies? Well, it's all done experimentally, and it involves dihybrid crosses and test crosses. So two concepts that you have already learned about to try to figure out these crossover frequencies, because you have to look at two genes at the same time, just like in dihybrid crosses. All right, so the first cross in this series is the same as Mendel's, parental generation to the F1 to F1 dihybrids. So we've got a pure homozygous dominant for both genes, and so that's also referred to as wild type, as I mentioned earlier. And then we have a pure homozygous recessive male. And the genes we're looking at here are black and vestigial. So black body and vestigial wings. Strange looking fruit fly, huh? But they really do exist. I mean, this is a real example. This is actually how they determine this. It's a real experiment. So the F1s, dihybrids, are all going to be the same. Heterozygous for both. That's what we always see in the F1 generation. They're always all hybrids, and in this case we're looking at two genes, so that means they're all dihybrids, heterozygous for both. But that's where we stop following Mendel's procedure. So now we're going to take an F1 female fly, heterozygous for both genes, and cross it with a male that is just like this male in the from the parental generation, but that means he's homozygous recessive for both genes, and that makes this a test cross. Hopefully you'll recognize this as a test cross, because when you do a test cross, you always cross an individual with an individual that is homozygous recessive. So it may be homozygous recessive for one gene, in this case homozygous recessive for both genes, two genes, but that's a test cross. So the second cross is not F1 to F2, it's a test cross, where a female dihybrid is crossed with a male homozygous recessive for both genes. So pure homozygous recessive, test cross, those are the gene symbols. Oh, and by the way, I forgot to mention that this is another way of using symbols. They're, instead of the capital and lowercase letters that we've been using up to this point, this is the way that most geneticists use symbols to represent wild with a plus sign, superscript plus, all lowercase, but superscript plus to represent wild, in other words, dominant. Wild is dominant. And then just all lowercase without the superscript plus to represent recessive. I guess they somehow found that superior to using capital and lowercase letters, probably because it's so hard sometimes to tell the difference between capital and lowercase letters when you're writing them out. So when we carry out this cross, we can predict, you can use a Punnett square, and that's what this is. This is a Punnett square that predicts the possible combinations of gametes in this cross. And because this is a test cross, even though these are two genes that we're looking at, there are really only 4 possible combinations because the male here is homozygous recessive for both genes. So we don't need a 16 square Punnett square. All we need is a 4 square Punnett square because there are only 4 possible combinations of gametes from the female, which is usual, normal, in a dihybrid cross, or from a dihybrid, I should say. But because the male can only produce one kind of gamete, and that's all recessive, that's all recessive, we really only have 4 possible combinations. So these are possible combinations of sperm and egg, and therefore possible results. In other words, what we would predict. But you'll notice in looking at these phenotypes that these 2 possible offspring are actually what we call parental types. In other words, they're the same as the parents. So these represent heterozygotes, which is like the parent, heterozygous for both genes, and then this guy or these flies represent the homozygous recessive for both genes parent. So these are parental types. Anything that are not parental types are the result of crossing over. So they're referred to as recombinants. They're the result of recombination and crossing over. Therefore, they're referred to as recombinants. So what we have down here are predicted results, these 2 rows here. Predicted results, the first one being if these genes are on a different chromosome. In other words, they assort completely independently, then we would expect to see an equal representation. In other words, 1/4, 1/4, 1/4, 1/4 of these 4 different possible phenotypes, both the parental and the recombinants. So 1 to 1 to 1 to 1 is what we would predict if these genes are assorting completely independently or on a different chromosome. If they're on the same chromosome and there's no recombination at all, that would be a recombination frequency of 0, then we would expect only parental types, no recombinants, because the recombination frequency is 0. So it'd be a 1 to 1 to 0 to 0 ratio. And that would be a situation where they are completely linked, 100% linked. What we have here are actual results. So when this experiment is actually done, when this cross is actually performed, these numbers represent what we actually see in the offspring. So what this indicates is that the gene is on the same chromosome because there are some recombinants. I'm sorry, there are some parental types along with recombinants. So because we're seeing both, this suggests that the genes are on the same chromosome. That means they're somewhat linked. They're not 100% linked, but they don't 100% assort independently either. They're somewhere in between. And so the idea now is to calculate what the frequency is. How, where in between completely linked and assorting independently are they? So we have to do a little bit of math here. But here's the actual ratios, 5 to 5 to 1 to 1. And how you determine that is to simply take the lowest number, 185, and make that 1. So 185 divided by 185 is 1, and 206 divided by 185 is pretty close to 1, and 944 divided by 185 is pretty close to 5, and 965 divided by 1 is pretty close to 5. So notice it's all pretty close, because, by random chance, you're not going to get exactly what you predict. But we need to find the frequency as a percentage, so the way we do that is to add up all the offspring, and that turns out to be 2,300 offspring. That's a lot of flies. And then we add up the number of recombinants, because what we're calculating here is the recombination frequency. So we add up the number of recombinants that we have in the offspring, and that's 391. And then we simply divide 391 by 2,300, and that gives us 0.17. And then we turn that into a percentage by multiplying by 100. So it's 17%, or 17 centimorgans, or 17 map units. So that tells us that these two genes, the black gene, the vestigial gene, are on the same chromosome, but they separate 17% of the time, which means they are linked. Then they're pretty strongly linked, because remember, 50% is the cutoff for being considered assorting independently and not being linked. So they are definitely linked. So because 17% is less than 50%, they are definitely linked. So to put it in words here, these genes are linked. Their recombination frequency is 17%, which is less than 50%. But although they are linked, the genes assort independently 17% of the time. They're separated by crossing over 17% of the time. So they're not 100% linked. And these genes are 17 centimorgans apart, or 17 map units apart, on chromosome number 2, which is the same chromosome we were looking at on the previous slide. So we can check our work by looking at the gene map on the previous slide. And when you do that, you're actually going to find that there's a 1.5% discrepancy between what we just calculated as the crossover frequency here and what it actually shows on that chromosome map. And the reason for the discrepancy, get ready for it here, this will blow your mind, but there's the possibility of double crossovers. So because of double crossover frequency, the distance is actually less than what we calculate. So what's going on there is that the genes are being separated by crossing over, but then they cross over back again. And there's a very low percentage of that possibility. It's only 1.5%, but it happens. And I think I made a mistake just a second ago when I said it was lower than expected. It's actually higher than what we calculated because of the double crossover frequency. So the genes are actually a little farther apart on the chromosome because of this double crossover frequency. It gets added to it. It gets added to what we calculated here. And that's it. It's a heck of a way to wind up this unit, but you do need to know that crossover frequencies, recombination frequencies to determine map units, what's used to do that is actual experimentation crossing with two genes at the same time, because we have to compare two genes and how far apart two genes are, how often they separate by crossing over. So it involves dihybrid crosses or using dihybrids, and it also involves test crossing. So that's really what you need to know and take away from this. And the whole concept of how the distance between genes on a chromosome are directly related to how frequently they separate from each other during crossing over or recombination. And that's it for this unit. There's quite a bit in this unit, so I suggest you study hard.